Hey, this is Dr. B, and let's have a look at some models for understanding medicinal chemistry and how to make it to the market. That's so from the theory all the way to the goal at the end. The receptor model is very popular for understanding drug activity. Even in the absence of adding a drug, the way the human system is believed to work is that you have a receptor, you have some sort of substance that's going to interact with that receptor. We'll just call that a ligand. And it will interact regiospecifically and even stereoselectivity with that active site, which is a simple way of saying we have a lock and key model for that ligand. It fits perfectly. It's forming favorable intermolecular forces. It's not reacting with it. It's just forming a nice key that goes in. And once it does that, it should produce a response. Fine. So there it goes. It produces a response. Now, if you're adding a drug, two things could happen. It could act just like the natural substance did, and we call that an agonist. It produces a response. Or it could simply block that receptor from producing a response. We call that an antagonist. Either one of these could become an effective medication. There are all sorts of other considerations. Let's say you've done that. Let's say you've made a antagonist for the TRPV1 receptor and it's effective against pain, for example. You better consider, if you're going to bring that forward, what the potential market cap is, which is the potential amount of money you could make in theory, right? This could be billions of dollars. Oftentimes, you better make sure you're dealing with something that's going to be available as a pill, since so few people would want an injectable with a needle. So almost all of these projects usually involve orally available medications. You better make sure that your lead compound is patentable, meaning that it is unique and hasn't already been patented. This leads to another very common approach, which is quote unquote ripping off a patent. Rather than try to start from the ground up, find something that's already effective and change the structure somewhat and come up with a different structure. For example, Achillean Pharmaceutical in New Haven, Connecticut has a drug which, oh boy, I think it was called elvucitabine, and it was not that different from AZT for the treatment of HIV. You might want to consider how far you're going to take this thing along, because if you take it all the way from idea to market, you may be dealing with a large percentage of, oh, I don't know, $1 billion. So if you're a small and nimble company, you might want to produce a promising lead and then just sell it, losing most of your market share, maybe all, hopefully not all, but you can move on. You won't break the bank. There are international considerations as well. The patentability in one country may be very different from the patentability in the other. The cost of your medication may determine where you can market it, and of course there's ethical considerations there as well. In terms of creation strategies, if you want to come up with a lead, you could just take a bunch of natural products and assay them against a receptor. These are typically done on things called like 96-well plates, and look for lighting up that assay, meaning hits. And that's a natural approach. This is not done hardly at all anymore. Usually they're taking synthetic drugs and doing that. You might think that the best way to develop a drug is in a rational method, and if that's the lock, let's make the key. And I agree that would be the logical way to do that, but it's oftentimes observed that it's much quicker to test everything than to try to do it by thinking about it. This may require a huge amount of investment for just a couple of medications, whereas oftentimes you can just test your whole library in a matter of days, even if it's thousands or more of substances. So just test everything. This is referred to as a combinatorial approach. There's combinatorial libraries that are just chemists who make thousands of substances, hopefully targeted, not just random, but that way you can test them against, hey, everything, right? And so this is a very effective approach medicinally. Once you do that, you're going to have to optimize your leads, and that's where a medicinal chemist spends most of its time. You'll do structure activity studies, SAR, to determine a structure activity relationship. That's where the R comes from. You'd like to maximize that potency and that selectivity, so it's very active, and it's also very inactive against other things, and it's also, hopefully, non-toxic and not too complex. That's what you're shooting for. Here's an example. This is a study that I was a part of where, yeah, I alluded before, we were dealing with pain medications, and here was a lead, okay? These CF3s don't look that promising. However, this is a substance that was, if you take a look at this data here, medicinal chemists call that a single nanomolar binder. That shows it's binding to the receptor site. And that is fantastic. That's something you shoot for. However, it better be solubility. So here, low numbers are good. Here, big numbers are good. You want it to be highly soluble and highly potent. Well, the potency is not bad at all, but the solubility is like nothing. This means you're making a brick. And if you're looking for that magic combination, perhaps the best lead is a 46 nanomolar binder, which may not be enough. However, it's plenty soluble. Really, the take-home message from this study is that there's no extremely potent soluble leads, so that's tough. Once you have your target substance, you celebrate, and it's time to run it through clinical trials. This means that you've not only gone through these in vitro studies, but you've also done probably some in vivo studies, meaning not just in the test tubes, but also in some animals you're seeing activity. And of course, there are ethical considerations there as well. All right, let's say you're going to market and you want to go through your clinical trials. First, you got to get through phase 1, might be up to 100 patients. The only thing you're really looking for is safety. It's well tolerated. The odds of passing that are pretty high. You've probably done enough studies already on your own that you know the thing seems to be fairly non-toxic. Now it's time for phase 2. This can involve up to 500 patients. It might take up to a couple years, but now you're looking for effectiveness for the most part, that it does what you want it to do. And since you're dealing with real human trials here, you're also going to be seeing if that safety is also maintained over a larger population. And this can involve 33% success. I think it might be even much lower than that, but that's what they're putting it as. So you've got a third, a 70% here, and if you've gone through at this point, if your stock is publicly traded, you're probably going to see a big bump at this point and some excitement. This means you can enter phase 3. It can involve up to thousands of patients. It can take another 4 years, but now you're dealing with very large populations and looking for anything that might pop out. And with this larger population, something very well might. They're putting pretty optimistic percentages here. I doubt they're really that high, but if you get through all of these things, you can get FDA approval in the United States, and you can market your drug, and this is not an easy thing to do. A given medicinal chemist perhaps only has a fairly low chance in his entire lifetime of creating something that does get FDA approved. However, this is the promised land, and this is a look at medicinal chemistry from the point of models and making it to the market.